So thank you very much for the invitation to be here. I am a card-carrying soil scientist and I'm going to talk about life underground. So I'm going to be talking about microbes. I'm a soil microbial ecologist, and it is what makes soil alive. But I'm also going to talk about relationships, and I'm going to talk about structure. And okay, so the winter of 2017 in California had the highest rainfall, the second highest rainfall in known history. Enormous rain after 5 years starting, that ended a year before that, this rainfall year, of incredible drought that made the news all over the world. So then we had the biggest rainfall, and you know, soil is, the properties of soil, the same properties actually can make it really resistant to drought, but also be able to handle floods, right? It's aggregate structure, right, that plays a big role in that. I drove by this field almost daily. I was fascinated by it. It's a field at University of California, Davis, managed by people who are on the staff. And I noticed, like, earlier in the year they had ripped the soil and it was too wet, and threw up these, what we call clods, right? It's just sitting there. It was just wondering where that would lead. And it led to almost 6 weeks of water sitting in the burrows, because it didn't drain rapidly, and then the next rain would come, fill up again. So it sat like that for a long time this winter, and I was cringing, but actually really fascinated to see how far it would go. And it did finally drain, and they're growing, I think, alfalfa on it right now. So I mean, life will go on. But, and this was not typical of all soils. I mean, other soils drain. This was a particularly bad one. So what's going on there? So this has a lot to do with the structure or lack of it in the soil. So in a well-structured soil, it's not a great picture, but you can see the white shapes in the left-hand side are aggregates, soil aggregates that have between them larger pores, and those pores are filled with either water or air. And in a well-structured soil, when that rain hits, when the deluge hits, that water will infiltrate and move on down to the groundwater. But in a poorly structured soil that has either compaction, that like crushes the big pores into smaller ones, or you don't have good aggregate structure, you're going to have a lot of small pores, and there the water will be retained at the surface, will flood above that. And in this case, we have this water table for that long period of time. So who's responsible for that structure? So this is the interesting thing about soil health, is that, or what soil quality, different words people use. You know, what is interesting to, what manifests itself are often these like physical properties, things like structure, the ability to drain. But the underpinning of that, I mean, who carries out a lot of this is microorganisms. They're responsible for a lot of what we accept as soil, or that we're used to interacting with this soil. They're underneath them. And here's a very famous diagram from Tisdale and Oates showing at different scales from the micron scale here, as we scale up to 0.03 millimeters, 0.3 to 3 millimeters. We have these different sort of levels of organization that make a bigger aggregate that you can see with your eye, right? But within it are smaller ones, within it are smaller, within it are smaller. And then you have different processes at play in creating this structure. And at a very small scale, we've got, you know, interactions between minerals and organic matter, microbial debris. We'll talk about that in a minute. And then at a larger scale, you are starting to glue together these particles into larger ones. And then we have polysaccharides, this extracellular polymers that all self-respecting microorganisms in the world have to be able to survive. We tend to select against it in the lab. We get rid of that sticky stuff because it messes up our ability to isolate microorganisms. But in the world, you need that. So that sticky stuff that they exude helps glue together these particles into these bigger bundles. And you also have, and I'm not scared by that because I saw already what happens. And you also have fungal hyphae that are ramifying, holding together these smaller particles into bigger bundles. Do I need to act a lot or something?
I'm not sure. I think we may be picking up some interference from space or
from space, right?
Probably CIA is listening in. I don't know.
Maybe my talk is so subversive.
Yeah, exactly.
And then we also have plants playing roles as well, building this greater structure. And soil organic matter is also really important to that. And I don't know if you know, but there's been a revolution in how we think about how soil organic matter is made. Get rid of your humic fulvic substances. That's all gone. And we know now, or we think we know now, that indeed organic matter inputs play a big role in it. You have plant residues like litter falling in it, but also rhizosphere inputs that are laundered by microbes so rather than having this resistant plant material particles hanging around that magically turn into soil organic matter, actually the microbes are breaking down this organic material and turning it into, and it's kind of going through them, right? This carbon is going through them, and they're turning it into microbes which die, and then they are the feedstock. Maybe 60–80% of the feedstock for making soil organic matter is dead microbes. So it completely changes how we think of how organic matter is made. By the way, we are still, trying to understand this. This is such a big change from how we used to think about how organic matter is made. It's interesting to think, okay, is the best thing to put glucose in the soil to make soil organic matter? So grow up lots of microbes and then have them die and make soil organic matter, rather than resistant carbon. Anyway, lots of things to think about. And so they are both enzymatic drivers, microbes, but also feedstock. And then you have this sorption, desorption. You need to bring the organic material together with the mineral fraction of soil. Because you're actually making organo-mineral matter. The way that organic matter persists is if it gets married to minerals and is sequestered and stabilized. Right? So it changes how we look at things. So, and this is the cycle of breaking down aggregates. When we do agriculture, we're often breaking down the big aggregates into smaller ones and leading to situations like that flooded soil. But you can also bring it back together, right? So we can build back up. You saw that continuum of aggregates. So managing the system to be able to then turn it around and bring back the structure of the soil. And how? Well, two things that you can do is you can feed the soil and don't abuse it as much. Don't physically abuse it as much. There are two things that you can do that will help to build aggregation. So this is an experiment looking at tillage. In California, no tillage is pretty rare because we irrigate and we have to move soil around and make beds and things. But there's interest in reducing tillage and looking for systems that might work. And I've been involved in this study for a number of years because I'm also talking about long-term research, which is really important for a lot of these questions. Long-term research plots for 14 years, where in a tomato cotton rotation, they looked at different treatments. So tilled ones versus not tilled ones with a cover crop or without a cover crop. So feeding the soil and not physically abusing it as much, right? Or sort of the treatments in a different language. And indeed we're able to bring back better aggregation and higher organic matter content. I'm not going to focus on that, but those indeed came back, although tillage is no-till is pretty complex about building soil organic matter. It is not straightforward. It isn't only about reducing tillage. In a lot of cases, reducing tillage doesn't lead to overall increases in soil organic matter. It's way more complex than people sometimes present it. I'm interested in the microbes. So, okay, this is typical sequencing 16S and 18S, and look at it's boring. You know, it's just all this like, okay, here's the different treatments at different depths, and oh, look at their, like, oh, different ones, there's these guys, and oh, there's not so many of those, and, you know, so it's typical sort of individual taxa-driven or taxa groups-driven sort of microbiology. We do a lot of work like this in microbiology, and I find it pretty boring, but it's like what you need to do these days is sequence. But anyway, we looked at what did I do? I skipped. Okay. We actually used a different approach, which is to take all that sequencing data, but then organize our groups, our bacteria, or our fungi into these using functional trait analysis. So that's bundling up these taxa into like, oh, you are the fast growers, you are the slow growers, you are the symbiotrophs, right, symbiotic organisms, you are the sapper ones, the ones that eat organic matter. It's trying to like deal with this complexity of what I showed you before of all these different taxa, like what do you do with that information, is to start to try to organize them into these groups. And this relates to what we were talking about yesterday, of who you're having relationships with, right? Which is really interesting in microbiology because there's so many choices of how you can relate to them. And here, and also that you need to look at all the other stuff going on in the soil as well, right? Which I'm not going to talk so much about the stuff in the bottom half. But in some things that we found in the no-till system was the bacteria. Look at the bottom green square. We had higher diversity and slower growing organisms, which we figured could be related to increased number of niches, physical niches in the no-till system, and reduced food access because you don't have the mixture of the organic matter throughout the whole soil profile. With fungi, we got more symbiotrophs, more symbiotic organisms that we figured was due to the lack of disturbing, beating up the soil. We need to start to create a relationship, you don't destroy it with tillage, but there being more stability for the symbionts. And with cover crops feeding, increased metabolic diversity due to increased carbon source diversity, because cover crops were like 6 different species. And also fungi looking at species diversity, that also increased more host diversity, both in plant hosts and also in organic matter materials. There's so much here that I'm just glossing over. I'm not going to go into detail, but I hope you can see it's a different way of looking at these data sets. Oops. And so taking stock, who are we engaging with? So assemblages, guilds, functional groups, communities, right, rather than individual taxa. People like smoking guns, they're like, oh, it's that one. It's getting away from that. But we don't know who is exactly building the structure. Who of those groups of organisms, who are the stickiest, the hypoliest, the most efficient ones in being able to carry out those processes? And also the scale is an issue. We could miss key insights because we bundled up, right? Big bundles of these where the action is happening on a really small scale. When we sample, we bundle up a whole bunch of different kinds of things going on that we can't really tease apart. And that's an issue as well. Okay, and that's just showing everything. Okay, infancy of indicators, right? We talk a lot about indicators. How can we measure whether our soil is in good shape? And Cornell Health Card is sort of one of the more developed ones. Purple is physical properties, yellow is chemical properties, green is biological processes. There really is very little biology in it. Respiration is there. And then carbon they bundle under biological, but you can see there isn't much biology there. So what are we missing that's essential? And, you know, again, do we need microbial biomass? Do we need diversity? These indicators of knowing where we are with our soils. So a number of questions. Okay, and then I want to end with Scott Park, and I hope I got 1 minute. He's a very successful organic farmer. He is my crush. And he's been farming for about 25 years as organic in a conventional pool of farms. And he started out as a conventional farmer. Farms about 1,500 acres, and he farms very like intuitive farmer, and he farms the below ground. The below ground is what he farms. So he says things like, don't disturb your soil biology, get out of the way. Don't overdo things. And you need to connect to the soil with your senses. Have to make long-term investments, it'll take time, but if you don't manage it below ground, you're gonna have problems. Take care of the soil and everything else will follow. So what happened to him with this flooding? So he grows tomatoes, and with tomatoes you have a contract with a processor, and they come and harvest on a certain date. And so that means you have to get the plants in ground on a certain date. And he was quite concerned about getting in because his soil was wet. But he was freaking out about bringing the planters on the field. But he was able to get his tomatoes in the ground about 1 day after the predicted planting date, whereas all around him, the farmers were 3-4 weeks late. Pretty much uniformly. So he definitely gained a lot by that. Communities of learners, I'll just end with this. So we need to like build communities of learners, practicers, with these continuums from farmers to the laboratory studies. Long-term experiments can possibly be a hub bringing people together. And I will end there.
Question based on, you passed very quickly on the revolution, but actually you use the soil is made of dead microbes. Did I get it right? That's the revolution?
That is part of the revolution, is that the feedstock is not directly, it's laundered by microbes before it becomes soil organic matter. So 60 maybe, there's different estimates of the actually material in the soil organic matter is former microbes, is microbe carcasses. Yeah. The other part of the revolution is that it's physical stabilization, that it's organic matter coming together with minerals. That was a little bit in old ideas, but it's more prominent now. Yeah.
I wanted to ask a question building on that. You said, so this figure of 60%, I'm sure that's a generalization, but that's pretty constant across soil types and uses, that 60% of soil organic matter is feedstock?
I don't know the answer to that one because the work has been done in fewer systems and it hasn't, gone out and been questioned everywhere. But I wouldn't be surprised. I mean, just the mechanism there is that. I mean, if you think of microbes, they're so small. They're at the scale where you're going to lay down the organic matter and connect it with the mineral, rather than big chunks of plant material. So, I mean, but I'm a soil microbiologist. I want them to be responsible and biased.
Right.
Yeah.
Okay.
Yeah.
During the experiment, did you use any pesticides like glyphosate, especially on the no-till plots?
On the no-till, absolutely. So, because no-till Roundup, right? People don't say no-till Roundup. They just say no-till, and then it feels so good because you're not beating up the soil. That experiment was done with Roundup, and there's been, so that could be embedded.
Is there any effect on the microbial, increase of microbial depth, for example?
Yeah, I mean, it doesn't seem to be getting in the way of there being big increases in microbial biomass when cover crops is added, when compost is there. And by the way, I just saw those fields. They're filled with critters. Even the no-till Roundup-ed ones are filled with worms and fauna that are often most sensitive to chemicals. But that's that system. Yeah.
How do you
communicate to the public this understanding, which it's not the conventional gardener understanding about feeding. So when you say feeding, a gardener, like an urban gardener, will immediately think, oh, you're putting fertilizer, you're putting sea salt, or you're putting
feeding the plants.
Whereas what you're actually talking about is the fruit exudates and the products of photosynthesis. So how do you deal with this confusion that when you use the word feeding, like how can we distinguish between?
Well, see, I think of it as shifting it to the microbes. They're the ones that are eating, right? So maybe you need to say feeding the microbes. And indeed, the exudates are part of it, but it's also you're putting in litter, or you might put compost in as well. There's like a whole bunch of different organic inputs you could use that would be on the platter of what you feed the soil. But I think what you do is you shift it from being only about the plants, and you shift it to the food web below that you're feeding. And I think that helps. Yeah.
I was thinking of this question of knowability, and so you just be looking on the very top of the taxa, and then you're trying to actually figure out what that means. And I was thinking, as if someone kind of took like an alien who took a kind of trolling net through the savannah, and it's like so the zebras and the bisons and the tigers and whatever, the humans and their cars, and they'll crush together, and then you figure out how it's supposed to work.
Yeah, absolutely.
So we would.
Or put you all in a blender, right?
Yeah, exactly. So an ecologist obviously has to, as you said, with the question of scale, can observe what's going on. What can a microbiologist observe? And how important is it to observe? Like, what are the ways to actually find out how we ever have this? And do you think we'll ever have the capacity?
And the question is, like, do you let that knowledge be in the hands of high priestesses who you send your samples to and they send you back some information about it and you trust that, or is it something the farmer needs to be able to do themselves? There's a push to use microscopy, which is, you can look at fungi versus bacteria, but it really gives you almost no information about your systems, but it's something that people can do, right? So I think that's less about data and maybe about building a relationship. And so it has its value for other reasons than the actual data that you get out of it. And then other people use indicators like fauna. Those are the big critter people. They're lucky you can see them, worms, fauna. And they would not exist unless there was underneath them, this pyramid of all this microorganisms as well. They can't live without that. So they're like a physical indicator of what you see. The more complex stuff, which is, this incredible diversity in soil microbiology and like what you do with that information. It's very easy now actually to get, just like you can get your DNA sequence, you can get soil sequence. But then you have this huge amount of data and information, which is really hard to make much out of. And I think that's where the science really has to go, is functional trait analysis, or these other sorts of approaches to try to pre-digest the information, or give a partly digested interpretation of it. And there probably will be services that do that. Yes?
I'm going to have to ask you to go with that question to the coffee break.
Okay.
Sorry. Thank you, Kate.